Well, welcome. Hopefully you came in. You are interested in learning how to care and feed your mongoose. If you are not, then you may have ended up in the wrong place. I'll give you a moment to leave the room. All right, now that they've left, the real people are back in. So welcome to the first video in Mr. Thompson's Anatomy and Physiology lecture series. We're kind of starting in the middle-ish of the course, jumping into some stuff because we'd already talked about anatomic vocabulary and position and stuff like that. Now we're going to get into some more basics. So we're going to be talking about macromolecules here. Biomolecules, you may have also heard them referred to, but we're talking about our nucleic acids, carbohydrates, lipids, and proteins. And so as we're going through, I have this slide deck posted if you want to have access to it. You can add it into your notes if you'd like. If you haven't done so, maybe pause the video and pull that up. It's going to be all basically the same slides, same information that I'm going to be using during this presentation. So we're going to be talking about the structure and the function of those different molecules, some examples, stuff like that. We're going to be coming back to these for biomolecules pretty frequently as we're talking about different organ systems and tissue types and stuff like that. So to start us off, to kind of give us a little bit of overview, what we're talking about are something called organic compounds, okay? And organic compounds are any compounds, chemicals, whatever you want to call them, that contain carbon, okay? Now back in the day, we were looking at substances that come from living organisms. That's why we call them organic. Now we have since come up with chemicals that have carbon but are not at all good for living organisms. Methane is one of them. However, it's produced by organisms, so maybe that's the bit. I don't know. Macromolecules, because if you remember from chemistry, molecules are just any atoms that are chemically bonded together, right? Macromolecules are large organic molecules, thousands and thousands of atoms, 3D shapes and stuff like that. So starting us off, we're talking about carbon. Carbon is an element. It is number 6 on the periodic table. Very versatile. It's got 4 electrons in its outer or valence shell. And if you don't remember what that is, you can ask me about that a little bit later. Maybe pull up your chemistry notes, something like that. It can form covalent bonds with up to 4 different atoms. CH4 is methane, so it's a carbon and 4 hydrogen atoms. So that makes it really flexible to create a whole lot of different types of bonds based on all the different elements that it can bond with. Now, most of the time what we're going to be dealing with is carbon, hydrogen, oxygen, nitrogen. Chon, I don't know if you remember, so like maybe from chemistry or biology you heard cho, chon, chomp. Those are just elements: carbon, hydrogen, oxygen, nitrogen. P in the case of nucleic acids is phosphorus. You have other elements that do that as well. Chlorine, for example, sometimes can be in there. So large organic molecules, also known as polymers, because they're typically made up of smaller building blocks called monomers. So think of like Legos or links in a chain or something like that. Some of our examples, the 4 that we're going to be talking about: carbohydrates, lipids, proteins, and nucleic acids. That's your DNA and your RNA, which I know you talked a lot about when you were a freshman. So we've got a couple of chemical reactions that we need to talk about. One of them is dehydration synthesis. Hydration meaning water. D means we are removing. So dehydrating something means you're removing the water. Synthesis is to combine 2 little things into one big thing. So what we're doing is we are taking bigger things, we're taking smaller things, making bigger things by removing water. It's also called the condensation reaction. So remember, you think back to chemistry, condensation is when water vapor in the air collects onto water droplets. That's what you get, the sweat on the outside of your ice water glass, that sort of thing. So basically, it kind of looks like this, okay? And so what we're doing, if you look here, they've removed the water, and so now we've got our subunits. Now these right here are our monomers, and then we've put them together to make our polymer. Now here's a little animation that shows how that works. So let me go back and, okay, so the carbon is black, the hydrogen is white, and the oxygen is red. And so basically what ends up happening is we've got our two monomers, and we've got a hydrogen and an oxygen are going to come out. And now if you notice right in here, there's our water, there's our dehydration because we've taken it out. And then where those atoms used to be now is a bond between an oxygen and a carbon. And so now we've created one larger molecule, that's sucrose, that's your table sugar, all right? So the other way then is hydrolysis. Hydrolysis, hydro meaning water, and lysis, to lyse something, L-Y-S, is to break it, split it open, or anything like that. So what we're doing is we are breaking things with water. So basically what we're doing is we're separating our monomers by putting that water back in. It's the reverse of the process that we just looked at. So here's our polymer right here, okay, our sucrose or our fatty acid or whatever it is, okay, and we're going to put the water back in. So now you can see here's the H2 and then there's the O, and so now we have two separate subunits, okay. We've lysed that chain, all right, we've broken it open. Here's another animation real quick that's going to show you basically the same thing. So here we have our bond in the green right there. A peptide bond is between proteins. Now we're going to put the water in there and break that bond and make two separate stable substances that are going to separate, and we'll use them to build other stuff. Two different amino acids. And so that's our hydrolysis. Dehydration synthesis is combining them by taking the water out. Hydrolysis is breaking them apart by putting the water back in. All right, so we've got our four examples now that we're going to be talking about, our four biomolecules. The first one is carbohydrates. Carbohydrates are small to large sugar molecules, right? So basically when we think about carbohydrates, these are the sugars. I guess you can think of it like that. Examples of carbs are monosaccharide, mono meaning one, we'll talk about that in a second, disaccharides, and polysaccharides. Those are the three different types of carbohydrates. A monosaccharide is one sugar unit, mono meaning one, saccharide meaning sugar, so one sugar. Examples of this is glucose. If you remember cellular respiration, you talked a lot about glucose, right? Deoxyribose, if you remember from DNA. Ribose is from RNA. Fructose is what's in fruit and vegetables and stuff like that gives them their sweetness. And galactose, which is important when you're breaking down lactose. Then we have monosaccharides. Well, no, those are still monosaccharides. And so if you're looking at the pictures, because you're going to have to know examples, definitions, and pictures, basically. And so if you look at this picture right here, your carbohydrates are going to be in a ring shape, okay? Most of the time it's going to be a hexagon. There are a couple of cases where it's a pentagon, but 9 times out of 10, if you see what looks like a hexagon like that, sort of a diamond-ish type shape, it's a carbohydrate, Okay, you put two monosaccharides together, and you end up with a disaccharide. Di is two of something. So disaccharide, two sugars. Your examples: sucrose, that's table sugar. Lactose, that's the stuff in milk. If you're lactose intolerant, you cannot break this stuff, and it gets your insides all messed up. Maltose is an important one. It's found in yeasts and stuff like that. We'll break it down. Humans, sometimes you'll get malt when you are breaking down starches and things like that when you eat them. Kind of sweet. It can be used as a sugar substitute in some cases. If you have two of those little diamond-looking things put together, that's your disaccharide. Okay. Also know your examples: sucrose, lactose, maltose. Another key that you want to sort of clue into when you're talking about different substances is how we name them will determine what it is we're talking about. So if you see OSE at the end, it is a carbohydrate. Okay. There's a couple other ones that we'll look at later where you can kind of use that same sort of how we name it tells us what type it is as a clue. Polysaccharides are many sugar units, so these are really, really big. There's three primary examples. One of them is cellulose in lettuce or corn. That's your fiber that you get. And it's basically long chains of glucoses stuck together. So it's very, very strong. You actually cannot digest cellulose, and animals cannot digest cellulose. They have bacteria within their intestines that does the digesting for them. Same thing with termites. We've also got glycogen. That is what animals use to store glucose for a future date. It kind of looks like a tree or a bush or something like that. And it's really easy to get to those glucoses so your cells can use those. Then we've got starch, which is typically just single chains or small branching chains of glucoses. And so this is the stuff you find in potatoes for like your French fries and stuff like that, in wheat, so your pastas and stuff like that are starches. Corn has a lot of starch in it also. It's one of the reasons why it's kind of sweet because of some of those sugars. All right, another one, lipids. These are compounds that are not soluble in water. They are hydrophobic. They can dissolve in hydrophobic solvents. So if you have lard, you can dissolve it in oil, but you can't dissolve it in water. Okay. The lipids, the biggest thing about lipids is they are primarily for energy storage. Okay. There's a few other things that they're used for, but primarily they are for energy storage. Okay. There's a bunch of different types of lipids. We've got fats, phospholipids, oils, waxes, steroids, and triglycerides. Now, steroids, we're not talking about the like ones where you inject them and you get like super swole. Okay, these are signaling hormones, and we'll talk about what that means towards the end of the semester. As far as how your body communicates with itself and stuff like that. So a few things we want to sort of keep in mind when we're talking about lipids. They've got 6 functions, primarily long-term energy storage. That's the big one. Please remember that. Highlight that. Protection against heat loss. Your skin has a layer of fat that helps you retain heat when you're cold. Protection against physical shock. Those are your shock absorbers, basically. If you didn't have any fat, it's padding to, I guess you can think about it as a certain extent. If you fall, that fat cushions your body and it cushions your organs and your bones and stuff like that. That's why if you've ever hit your shin on something or the bony part of your elbow, right, it seems like it hurts way more than it really should because you're hitting that bone directly. Where other places like your arm or your leg, you've got those soft tissues to absorb that impact, so it's not transmitted to some of those other deeper tissues. Protection against water loss, because it's hydrophobic, that means the water cannot pass through it. They're not going to mix. Oil and water don't mix, right? You put vegetable oil and water together and they separate. And so you've got that barrier, so the water can't come out. Also, chemical messengers, your hormones, the steroid hormones that we had talked about. And the phospholipids are the major component of the plasma membranes, which we'll talk about here in about a week or so when we talk about cells and transport and stuff like that. So subunits of lipids, there are not really any. I suppose if you were going to have one, you would talk about triglycerides. Triglycerides have a glycerol and 3 fatty acids. And so it looks like this. So here's the glycerol, right? It's sort of the backbone of it. And then the 3 fatty acids, tri meaning 3, okay, sort of hang off like tails. And so I always think of a jellyfish, so you've got your triglyceride right here. And then here are your fatty acids just sort of hanging off, and it's sort of floating around in your body and stuff like that. Different fatty acids, if you've looked at food labels and stuff like that, you've probably noticed that there's 2 main ones. If you've got saturated fatty acids, those are the bad ones. They don't have any double bonds on those chains. Unsaturated fatty acids are good. They do have double bonds, and so basically what ends up happening is they're not able to sort of fit together and solidify, and so they're a little bit better for you. Here's what a saturated fatty acid, and when I talk about the double bonds, I'm talking about in between the carbons right here. And so basically, if I can sort of step back for a little bit, if I were to take this chain right here, okay, and sort of expand it so I could see all of the atoms, it would look like this. So here's my chain of carbons, and then I've got hydrogens hanging off the end there, right? So I've got all these hydrogens, and these are all single bonds right there, okay? So that is a saturated fatty acid. It's saturated with hydrogens, if you're interested in knowing. So we put all the hydrogens on there that we can, okay? Unsaturated then is I remove some of those hydrogens and then I replace them with double bonds, okay? So let me get rid of some of those hydrogens. And then what I'm going to do is I'm going to put double bonds in here for those hydrogens. Okay, and so what we end up with, here I'll show you a picture, is something with kinks in it. And so you can have mono unsaturated, you can have poly unsaturated. That just means it's got a lot of these bonds. And these are the ones that are better for you. Saturated fatty acids are going to be like your animal fats and lard and stuff like that. Your unsaturated fatty acids are going to be your vegetable oils, peanut oil, canola oil, that sort of thing. Moving on, we have proteins. Proteins, also known as polypeptides, are made up of amino acids. And in animals, there are 20 different amino acids. They're held together by something called a peptide bond. So if you hear peptide, you want to know you're thinking about proteins. If you hear amino acids, you want to know you're thinking about proteins. These are things, when I give you these hints, these are things I recommend you write down to help you remember this stuff. Because one of the things, I'm sure I mentioned it, there's a lot of vocabulary. I'm going to be throwing a lot of new words at you, but a lot of them, they have things in common. They have roots and suffix. And so if you can sort of tease those things apart, it'll help you a lot going through this. So you want to pick up those keywords and highlight them as you're taking notes and stuff like that. Okay. The functions of proteins, there's 6 main ones. Storage, so albumin, like in egg whites, is energy storage. Transport, hemoglobin is a very important protein that we'll talk about in January. Regulatory, there are also certain classes of hormones that are proteins, even as small as like individual amino acids and stuff like that. And we'll talk about that when we do the endocrine system. Movement, your muscles are made out of protein. As a matter of fact, for the most part, it's made up of 2 proteins, and we'll talk about that October-ish. That's actually the 2nd or 3rd major organ system unit that we talk about. Structural, your cellular membranes, there's a lot of proteins in there. We're going to talk about those when we talk about transport. Your hair and your nails are both made up of protein. They're not alive. So don't let the shampoo manufacturers tell you otherwise. Your hair is not alive. However, there is, it can be healthy because it's just protein chains, basically. What else do we got? Enzymes. Cellular reactions are sped up way faster than they would happen normally because of enzymes. So those are all really helpful. We'll talk about enzymes a little bit as we need to, but we're not going to spend a whole lot of time talking about proteins and stuff like that. Proteins are really complicated in that they have different levels of structure based on how they're arranged and stuff like that. So the first one is primary, then after that is secondary, tertiary, quaternary. And one thing to keep in mind is each structure builds off of the next one. You'll see that here in just a second. So primary structure, that's just the line of peptides. Typically this is a straight chain of them, okay? So I've got all the amino acids just sort of stuck together, right? If you remember from biology, you talked about transcription, translation, protein synthesis, all big scary words, all big scary processes. This would be the protein synthesis part, okay? We've already gotten the amino acids now that we put them together to make our primary structure. Now, once you get a couple of chains together, you have the secondary structure. Typically, well, there's two kinds. There's coils and there's pleats. Also known as the alpha helix, so it looks like a spring, and you have bonds that will attach at certain points of the amino acid. I don't think we really talk that much about like functional groups and stuff like that, but basically you have bonds forming within different parts of the chain. That's what these dotted lines are right here. So you've got alpha helix, and then the other one is the beta pleated sheet. So if you took a piece of paper and you would have folded it like an accordion, that's the beta pleated sheets. So that's secondary structure. A series of chains that you have sort of bonded together, and now they've got a physical shape. Tertiary structure is you're getting one or more secondary structures along with primary structures, and you're putting them together to create a larger 3D shape, basically. You've got hydrogen bonds, ionic bonds, disulfide bridges, which is just sulfur on sulfur, which has to do with functional groups, which I didn't really talk about. If you're interested in looking at a little bit more about functional groups and digging deeper into how the bonds form and stuff like that, let me know, and we can have a chat a little bit more about that sort of thing. Your tertiary structure are known as your subunits. In a lot of proteins, this is basically what the protein is, or the completed protein where you will take multiple subunits and put them together, and then they work together. So honestly, if you kind of see something that looks like this, that's your tertiary, okay? Quaternary is multiple subunits, but hold on, I skipped something. So you can see in this one, you can see that here's an alpha helix. That's a secondary structure, but you've got other structures here because there's your beta pleated sheet. And then you've got other helixes and pleats and stuff like that put together. Quaternary is multiple subunits being put together, two or more. They're typically globular in shape. So if you look at something that kind of looks like two or more blobs put together, that's your quaternary. Aqueous, they're formed in an aqueous environment. Aqueous means it's dissolved in water, okay? And so one of the things that it's understood is your body, your internal body is an aqueous environment. And so basically what your body is attempting to do is try to keep everything in that liquid environment that all life arose from. Basically what we had to do is we had to figure out evolution or whoever. So the major problem to overcome was how do you bring the ocean with you when you go on land? And so our internal environment is aqueous. Your bigger enzymes are quaternary. Hemoglobin is quaternary. There's 4 subunits clustered around an iron molecule. Kind of looks like that, basically. And the iron molecule sits in the middle here. I think we've got one more. Nucleic acids. Nucleic acids are super famous. There's only 2 types. DNA, which is deoxyribonucleic acid, that's your double helix, your staircase, and then you've got RNA, which is a single strand ribonucleic acid. Nucleic acids are composed of long lengths of nucleotides that are put together by dehydration synthesis. Now remember, we talked about this. Dehydration synthesis, we are dehydrating, so we are removing water. And then the space that's left snaps together into a bond to create a larger molecule. We're synthesizing a larger molecule. I think we talk about functions here. Not yet. So we're going to talk about the nucleotide. There's 3 parts: the phosphate, the pentose sugar, and the nitrogen base. Nitrogen bases, remember, A, T, G, and C, adenine, thymine, guanine, and cytosine. In RNA, it's uracil instead of thymine. I'm sure to many people's relief, we're not going to be talking about codons and rating the charts and figuring out like what is AUG, methionine, maybe, I don't remember. We're not going to be doing all of that stuff, okay? And base pairings and when you're doing DNA replication and stuff like that. We don't really talk about that in here, which is unfortunate because I think it's all kind of cool, but I guess that's fair. I'm a science teacher. So here's what it looks like. You've got your phosphate group. There's your sugar. If it's deoxyribose, it's DNA. If it's ribose, it's RNA. Your nitrogen base, which is A, T, G, or C. Some people think it looks like a chicken. If that helps, go for it. Then here's our double helix, and the bases are in the middle. They're held together by hydrogen bonds. Well, shoot. I guess that's basically it. Function, it didn't really mention it on here. Function is transmitting information, right? The whole instructions to how to make you, and cats, and bacteria, and blue bonnets is all in the DNA. And then RNA, they kind of do the work. You've got the messenger RNA and the transport RNA and all that other stuff, right? They run the instructions for the DNA in order to make the proteins, and then the proteins sort of go off and do all the mindless heavy lifting and stuff like that. So, that's basically it for macromolecules. We got 4 kinds, right? Proteins, carbohydrates, lipids, DNA, nucleic acids. So things that you need to make sure that you can do, okay? You need to know names. You need to know functions. You need to know examples. Okay, so go back through and look at examples. Name examples like sucrose, but also structure examples like double helix, that sort of thing. Know the characteristics, because I may say, all right, this macromolecule has primary structure, go. That's proteins, right? That's the only one we talked about, right? This other one is primarily for long-term massive energy storage, go. Hope you said lipids, because that's the right answer. Okay, so those are the sort of things I'm going to keep you, you're going to be looking for. So when you're going back through and you look at your notes, you want to make sure you've got some of that stuff in there. If you have any questions, let me know. I'll see you in class. I'll see you on Zoom. Send me a message. Send me an email. That sort of thing. We'll work our way through it. Thank you very much.